Okay, this is a video lecture for Chem 255. It's the midterm review for Introduction to Biochemistry at St. Francis Xavier University. This review will be quite comparison to the material that we've covered so far. By no means will this be enough for you to study for the midterm. I suggest that you use it as a supplement and a reminder of the important points that we've talked about so far. And so if we bring everything that we've talked about back to the first day of class, we have to understand that we're talking about all of these issues in the context of energy flows within a biosphere. We're trying to understand the mechanisms that we have in place that allow us to tap into this energy flow. So the plants and animals that are around us have developed ways to harness energy and create ordered molecules that eventually will break down and the energy stored in them will be released. We've simply inserted ourselves into that energy flow. We've found a way that we can use these complex molecules. We can release the energy that's stored inside, and we can direct it in the cell, in tissues, in the body, so that we can harness and use it to our own ends. We exploit that energy so that we can stay alive, so that we can develop and grow and reproduce. So the energy flow is already there. We've simply found a way that we can take these food molecules, break them down to their constituent products, releasing energy, and then if we do that in a way where we can focus that energy and capture that energy, then we can use it to do work. The way that we do that is to adhere to the two laws of thermodynamics. We express these laws in terms of Gibbs free energy. This is an expression that takes into account enthalpy and entropy, which describe the two laws of thermodynamics. And the net change in Gibbs free energy allows us to determine if a process is likely to occur, if it's spontaneous, if it requires any extra energy, or if it can give off energy. And so it's important to understand this principle, since what we're trying to do is to harness the energy from our surroundings. So Gibbs free energy is a number that identifies the change in free energy for a given reaction. It identifies specifically how many kilojoules are being released per mole of substrates and products in that reaction. We think of it as the ability or the energy available to do usable work. If a reaction or a process has a negative delta G, that is, it releases kilojoules, then it's called exergonic. We think of that as spontaneous. It's more likely to occur. If it's positive, that indicates that it requires the input of energy, and that's something that won't occur on its own. Free energy is not the only way that we describe the likelihood of reactions in the body, because delta G is always changing. Delta G will describe whether or not a reaction will proceed on its own, but if we try to assess it in the body, it's impossible to peg down one value for Gibbs free energy, because it's subject to the concentration of substrates and products for that reaction or for that process. So in that case, we have to introduce a new term, Gibbs standard free energy. And this is just a measure of the likelihood of a reaction at standard temperatures and pressures, standard concentrations of substrates and products. So we remove all variability in the system, and then the nature of the reaction is described over here by Gibbs standard free energy. This is delta G naught prime. This is standard free energy that we refer to in biochemistry. This term over here inputs those variable factors back into the system. So we can compare reactions using the nature of the reaction, and then we can assess how that reaction will behave in a real living system if we know the gas constant, which is always the same, if we know temperature, and then if we know the concentration of products and substrates for this simplified reaction here, A converted to B. So this is simply a way that we can get a handle on the spontaneity of the system without being inside the system and without knowing the conditions that are inherent to that system at any point in time. It removes that variability. Now, knowing how reactions are likely to occur is great. We do need that as one of the foundational principles in understanding metabolism. We also have to know where metabolism occurs. And it occurs in an aqueous environment inside the cells, surrounded, all the metabolites, enzymes, pieces of us, pieces of the cell, are all surrounded by water. And water is one of the main factors that drives proper formation of proteins and, in some cases, reactions themselves. Water, we've talked about, has special properties. It's a polar molecule where there's unequal sharing of electrons between the hydrogen atoms and the oxygen atoms. And so the hydrogens on water tend to be partially charged positive, and the oxygen atoms tend to have a partial negative charge. So there are two poles. They're not completely charged, they're partial charges. These poles will interact with other water molecules, they'll interact with other polar molecules, and even other charged molecules. And so the partial charges will attract the opposite charge, as shown here. The positive hydrogen ion is attracted through a hydrogen bond, these three dots, to the negative partial charge on this oxygen atom here. And this is one of the main driving factors that allows metabolism to occur. So we talked about how water can influence shape and structure. Not only does it interact with polar molecules, but it, quote unquote, interacts with nonpolar molecules by not interacting with them. And that might sound immediately complicated, but it can form polar molecules by the absence of hydrogen bonding to those nonpolar molecules. Water is always trying to search for hydrogen bonds or for charged molecules that it can bind with. When it doesn't find those in nonpolar solutes, it's forced to look elsewhere. And where it looks is immediately next to that water molecule, to the neighboring water molecule. And all of a sudden, we create this ordered sphere around the nonpolar molecule. The inability of water to bind to that molecule forces it to bind to other water molecules, and we have what's called a hydration shell that forms around this nonpolar solute. This is why these molecules tend to aggregate in clumps or in balls. Fat will tend to end up in a micelle or a bilayer because the nonpolar part is pushed towards the center. And this action of pushing it into a spherical shape maximizes the entropy in the system. It returns it to the natural state, or as natural as it can be in a given situation. Water also provides the medium inside which all of metabolism occurs. And this is important because water tends to ionize partly on its own, and the ionizing factors, the hydroxyl groups and the hydrogen atoms, will tend to act in a lot of metabolic reactions as well. Now, how water ionizes is described by the dissociation constant, or the pK. Water itself it doesn't tend to ionize very much, but some substances do ionize more. Some substances ionize completely, like hydrochloric acid, completely ionizes and causes a spike in hydrogen atoms inside the cell, which can wreak havoc with the normal operation of the machinery in the cell and cause proteins to unravel and things to break down. That's on the other end of the spectrum. In the middle, we have what are called weak acids. These are things that tend to dissociate, but they don't dissociate completely. Oftentimes, you'll find them half in their acidic form and half in their conjugate base form. And this is important, especially in the body around a neutral pH, because these molecules will act as proton donors or acceptors. They will buffer changes in pH inside the cell. We don't want large changes in pH. We want small changes in pH, and these collective molecules will ensure that no large changes in pH occur, no excessively large changes in pH. So we can see a couple of examples here of molecules that will dissociate in response to a change in pH. We've also learned since this that amino acids themselves also serve to act as weak acids. We will talk about this a little bit later on in the video, but we have a general rule that any pH more than 1 unit above the pKa of a weak acid will mean that the proton is off of that weak acid. Anything above this top blue line here. And then anything below 1 pH unit below the pKa will mean that the proton is on, so that this is fully associated. And this blue region, the region between those 2 points, is what's considered the buffering region. It's the region where we see this flattened pH response. Whenever we see a flattened pH response, we know that buffering is occurring, but there's only a given range that any one buffer can work within. No one buffer is going to be able to buffer all of the changes in pH within the cell. So we understand some basic foundational concepts with respect to the environment and how metabolism has to work. And now we need to put that into context of metabolism. It's an inherently complex series of reactions. There are so many overlapping reactions. It seems to be quite disordered, but we'll see in the second half of the course it's not really the case. We're just trying to decipher how all of these things fit together. Now we mentioned that free energy is always changing in the cell, and it's that way for a reason. We need to be able, in some cases, to push a reaction forward or pull a reaction backwards. We need to change the free energy of that reaction. And we can do that because the free energy of a reaction is influenced by the concentration of substrates and products. You can think of it as a dam holding back the flow of a river. If there hasn't been much water building up behind the dam, there isn't much pressure for the river to flow forwards. But if you have a large volume of water behind the dam, then there will be a large pressure pushing the river forward. And we can harness that to do work, like making energy by passing it through turbines. Here it's a similar situation where if we have a lot of substrates, that will push the reaction forward. So we've made up a couple rules. We figured that any small positive delta G, any small positive standard free energy, just related to the nature of the reaction, can be overruled. It can be influenced by substrates and products. But large delta G's cannot. Large delta G's can't be pushed one way or the other if the nature of the reaction is just so impossibly strong in one direction or the other that it won't be influenced by the substrates and products. We can push a reaction forward if we have more substrates. We can push it backwards if we have more products. And what we call the division between small and large is around 14 kilojoules per mole, plus or minus 14 kilojoules per mole. So a quick summary of the rules that we have are large positive and large negative delta G's are irreversible, Negative likes to move forward spontaneously. Positive is non-spontaneous. It won't move forward on its own. In fact, it would like to move in reverse. Small delta G's are reversible. Small negative delta G's will tend to move forward. Small positive delta G's will tend to move away from the creation of products, but it can easily be pushed in the direction of products by accumulating substrates for that reaction. Now, the process of releasing energy must be managed acutely. We can't just take these complex molecules that we get from our surroundings and then combust them and hope that the energy will go to some usable process. We have to direct that energy. That's the job of metabolism, is to direct that energy. If we just combusted one molecule of glucose, we get a large amount of energy released, but without any way to direct that energy, it's very likely to be damaging to the cell. So we collect that energy in packets, high energy compounds that we can then use in different places in the cell to deliver that energy to do work. The high energy molecule of choice is ATP. We build ATP from ADP, from AMP, and simply from adenosine by adding multiple phosphates. And these bonds are hard to make. They're not easily made. They take an input of energy to make them. But once we have these bonds made, they stored that energy until we break those bonds. The phosphoanhydride bonds we talked about as being more unstable, and so they are more energetic and release more energy when they're broken. The phosphoester bond is more stable, so it doesn't release as much energy. But this is generally how we transport energy in the cell in terms of ATP. When we're exercising, we use a lot of ATP. When we're recovering or resting, any ATP that has been broken down is resynthesized for future use. So ATP generally we try to maintain ATP at a constant level in the cell because it is so versatile and we can use it for so many reactions. The way that we make ATP, and really the way that we harness energy from the surroundings, is by ripping off energetic electrons from those complex molecules and delivering them to oxygen. We'll see exactly how that works, but in the process of transferring those electrons, we end up harnessing energy from the electrons that we use to make ATP. So carrying these electrons is a very important job in the cell. And we have two main electron carriers. The first one shown here, nicotinamide adenine dinucleotide, or simply NAD for short. We see NAD here. A different version, NADP, simply has a phosphate group attached to it. And these will harness, they'll add electrons to this ring shape here and deliver them to the electron transport chain, which we haven't talked about yet, but it's an endpoint in metabolism. So these are things that we're always constantly loading with electrons that then ferry them to their endpoint and release them in order to harness their energy to do work. The other main electron carrier is flavin adenine dinucleotide. You'll see both of these are made of adenosine. They have a phosphate bridge and a slightly different vitamin derivative that does the actual electron carrying. Here riboflavin does the electron carrying, where niacin did it in the previous example. And so with this in mind, we have all of the major ingredients in place to start to understand the infrastructure of metabolism. What we've outlined so far are the rules that metabolism has to work by, but now we're progressing on to the infrastructure that supports metabolism. And in the big picture, this infrastructure is largely supported by proteins. Proteins are the infrastructure that do work in the cell, that support metabolism. And so we've spent a lot of time understanding where proteins come from, how that information is transmitted between generations, how proteins are made, how they might be shaped, why they're shaped differently, all because we need to understand the infrastructure for how metabolism works. So we spend a lot of time talking about DNA because these molecules, the DNA molecules, will contain the information needed to make every single protein in every single cell in every single tissue of your body. That information has to be read, it has to be maintained, it has to be understood. We have to be able to interpret it and then turn that into something usable, into a protein, because DNA itself is not a protein. We do that by understanding the sequence of molecules inside DNA and translating that into protein structure. Proteins are built by constituent amino acids being linked together. Each amino acid is different. It will drive the individual structure and function of a protein. So we'll go quickly through a couple examples, a couple things that we talked about. This is the more recent material, so not something that I'm expecting you need too much of a refresher on. But you guys have known for a long time and have probably been reminded of this constantly that the instructions to make protein are all based on 4 main molecules. They're all types of deoxynucleotides where the base is slightly different. And the sequence of these nucleotide bases in a DNA strand will inform, will dictate the sequence of amino acids in a protein. We store DNA as a double-stranded or a two-stranded, a double helix. When we need information to make new proteins, we don't directly access and then make the protein from DNA, but we make it from an intermediate molecule called RNA, ribonucleic acid. Something that is a little bit less stable, but a really interesting molecule because it seems to have its own catalytic or enzymatic activity. It seems to interact with both DNA and proteins. Now, the system as it's set up now recognizes 3 base codons. So every 3 bases represents 1 amino acid. There's lots of redundancy, and based on this code, we will link amino acids together through the actions of tRNA molecules, or transfer RNAs. The idea being that only specific tRNAs will bind, carrying specific amino acids depending on the code found in mRNA. Now there's a lot of complicated genetic information in this section. The actual nucleotides themselves seem very complex too. It's difficult to understand how they might have ever been synthesized. We have proteins that do that now, but before we ever had proteins, how would it possibly have come to pass that we made nucleotides? And so we saw an example of how 3 very basic ingredients right now are the only things that we need to start making purine nucleotides: aspartate, glutamine, and then bicarb. These are things that would have been available on early Earth as well. And with enough energy, we saw that these products do sometimes spontaneously form. So what seemed like a giant leap in understanding of where these came from might not be as inexplicable as we once thought. Every single standard amino acid that we use to make proteins has 4 groups bound to a central carbon. The central carbon is called the alpha carbon group. On the left there's the alpha amino group. On the right the alpha carboxyl. Down below is just a hydrogen atom. And then above is the R group, or the side chain. And it's here where every amino acid is different. And side chains can be big, can be small, can be charged, can be completely nonpolar. And it's the characteristics of the side chains that give amino acids their characteristics. Now, in solution by themselves, these amino acids act as weak acids. The amino and the carboxyl groups ionize. Some R groups ionize. And so these amino acids will tend to buffer protons in the cell. Remember, the pKa, the dissociation constant, identifies the point where the acid form and the conjugate base form are balanced. And so if we move away from the pKa, we move more towards one of those forms. We also went through an example of calculating the isoelectric point for an amino acid. In general, in the cell, amino groups will have a positive charge while carboxyl groups will have a negative charge. Those charges offset, but any charge on an R group on a side chain will influence the charge of the amino acid. Additionally, the pH of the solution containing that amino acid will affect the total charge on that amino acid. It's all based on what the pKa's of those individual functional groups are and in what pH they are sitting currently. So free amino acids we've talked about are weak acids. Amino acids have specific charges. R groups might be charged. When we link amino acids together in a protein, then, proteins themselves can also be charged. Calculating the charge for a protein, though, is more simple, or not as complicated as calculating the charge for every individual amino acid, because when we link amino acids together, alpha aminos of one bind to the alpha carboxyl of the next, and those two groups are now not available to ionize. They won't carry a charge. And so in a protein, there will only be one alpha amino group, one alpha carboxyl group, no matter how long the protein, and then any R groups, any side chains that ionize, that will contribute to the charge of that protein. Again, we talked about if the pH is more than 1 unit below the pKa of a given group, we can assume the proton is on that group. And if the pH is 1 unit above the pKa, we can safely assume the proton is off. Now, primary structure of proteins is directly pulled from the sequence of nucleotides in mRNA, and the primary structure of proteins informs all other levels of structure in a protein. It's the sequence of amino acids that gives it the shape that it has, that results in some standard secondary shapes, alpha helices and beta threads or beta sheets. It's the sequence of amino acids in a peptide that tell us whether or not we're going to have a turn, whether or not we're going to be able to loop alpha helices together or make beta strands turn around and interact with each other. We often find proline and glycine residues in turns. When we start to put secondary structures together, super secondary structures are what we call motifs. And then when we group motifs together, generally this is the intact, fully functional protein. The tertiary structure of a protein is generally the functional unit of that protein. Now, some proteins will have to aggregate. Some proteins are called multi-subunit proteins, and so it might take multiple polypeptides in order to make that protein function, but then some proteins will stay in this tertiary structure as well. All of this stems from the primary sequence of amino acids in a polypeptide chain. When we start putting these things together, we've seen that secondary structure tends to occur naturally on its own. We haven't talked a lot about how proteins are folded. The rest of the process is mediated by other proteins, but we won't get into too much detail there. We do see some fantastically intricate shapes that occur, and the shape dictates function. One really cool example of that is with this 8-stranded beta barrel, where the internal area in that barrel is generally hydrophobic. And so nonpolar molecules tend to be stored inside this barrel, transported around the cell, and manipulated as such. If an enzyme requires the binding or the transport of nonpolar molecules, they might have a structure like this to accomplish that goal. This is retinol binding protein, but fatty acid binding protein contains one of these barrels, and its job is to move very nonpolar fatty acid chains around the cell to be metabolized for energy. Tertiary structures are fully functional enzymes, and while they tend to all aggregate together in one unit, one subunit, sometimes we'll see different domains. Different domains are generally independently functional areas of a protein, but they're still part of the same polypeptide chain. They're not different subunits, they're just different groups, different functional groups within the same protein. And so this example, glyceraldehyde 3-phosphate dehydrogenase, has two jobs. It has to bind glyceraldehyde 3-phosphate, and that's the job of one subunit. The other subunit has to bind NAD so that it can be loaded with electrons from glyceraldehyde 3-phosphate. Two different jobs, two different domains within the same protein that allow that job to occur. Notice the shape of these two domains, specifically this NAD binding domain. We've started to notice that some sequences tend to be related to specific functions. So here this beta thread, alpha helix, beta, alpha, beta, alternating repeating structure, we've noticed tends to be associated with binding nucleotides. We've actually made a lot of advances in this area where now if we know the sequence of a protein, we can guess what shape that protein will have, and then we can guess what function that protein will have from the shapes and from the commonly repeated structures that we expect it to have. We can do all of this with computer models now. Here, this example of really cold active citrate synthase, an important enzyme in metabolism, where these two proteins, the top being cold citrate synthase, the bottom being citrate synthase from a hotter environment, these two proteins have nearly identical shapes so that they can carry out the same function, but the sequence of these proteins is very different. Here, these two are only 27% similar. 73% of their primary amino acid sequence is different. And so you would expect that these are completely different proteins, but they're not. You can see the shape is very similar, so we expect they have very similar functions. And in fact, this cold protein has adapted to make the reaction easier to manage by opening up the active site compared to the hotter environment citrate synthase. The surface is more charged. Here it's more negatively charged, as shown in red. This, we think, facilitates interaction with water. And we also find that in the sequence there's much less proline, which we've already identified as a rigid amino acid. And so having less proline would make the protein more flexible. Very important for a protein that has to work in a cold environment. And so proper protein folding is of vital importance. Proper protein function is of vital importance in metabolism. And when these things go wrong, sometimes metabolism won't just stop working, but we can actually cause severe health detriments by simply changing the shape or the size of one small protein. And one example of that was the progression of Alzheimer's disease and some other diseases that we'll come back to, all result from accumulation of beta amyloid proteins. Proteins that we make normally, that we think are part of supporting normal memory function and filtering out unnecessary memories. These are things that are always being produced, but when they're produced improperly, it can result in severe health detriments or consequences. Here specifically, the problem is that beta amyloid proteins are cut a little bit too long. And when they're cut a little bit too long, they tend to aggregate and they interrupt normal neuronal function. Here, too long is only 42 amino acids versus the normal length of 40 amino acids. And so while we're still learning a lot about how and why this disease progresses and how we can treat it, it's a very acute and striking example of how protein structure and function are tightly linked, and how with improper structure we can get improper and negative function. So I hope that this video has helped you grasp some of the main topics that we've discussed at length in class. It certainly won't cover everything that we've come back to repeatedly. There are some things that we tend to talk about a lot, and those are generally important points to consider for the midterm. If you have any questions as you go through this lecture or as you go through any of the notes, please feel free to email me. I'm happy to help you out, and otherwise I will see you at the midterm.